Hello friends, in this video we'll be discussing about prokaryotic cells, which is an extension of the cell biology discussion. So prokaryotic cells include three important groups of organisms. The first one is bacteria, second one is cyanobacteria, which is also known as blue-green algae, and the third one is mycoplasma, which is also called as PPLO, pleuropneumonia-like organisms. These are the smallest known organisms which lack cell wall. So let's talk about prokaryotic cells in general. They are smaller and multiply more rapidly in comparison to eukaryotic cells. They have small size and rapid multiplication rate, and they also vary greatly in shape and size. They have different types of shapes and sizes. Now let's compare the four basic shapes of bacteria as per mentioned in NCERT also. This is the bacillus, which is rod like bacteria, which is given in the diagram also, which has spores. Spores are present in all bacteria. Spores are the structures which have thick cell wall, which prevents them from dying in extremes of climate, and when the climate or the environment condition gets normal, the spores germinate to form other bacteria, more bacteria. The second shape is spherical shape, and the bacteria which have spherical shapes are called as coccus singular and cocci when they are plural. So second shape is spherical. Third shape is comma shaped, which is mentioned as vibrio. The bacteria which have comma shaped are called as vibrio, and the fourth shape is spiral, and the bacteria which have spiral shape are called as spirilla when they are plural and spirillum when they are singular. Spirillum also have flagella or flagellum. Flagellum is for singular, and they have flagella on both of their ends. We'll discuss about their structure, their flagella in details. So for the time being, let's start the discussion. The organization in prokaryotic cells is fundamentally similar. All prokaryotic cells are fundamentally similar, even though prokaryotes exhibit wide variety of shapes and functions. But being prokaryotes, they have fundamental similarity in themselves. Now let's talk about the cell wall of prokaryotes. Cell wall. All prokaryotic cells, all prokaryotes have a cell wall around them, around the cell membrane. They have cell wall. The exception to this is mycoplasma, which are the smallest living organisms, and they lack cell wall. They completely lack cell wall. So for prokaryotes, we have studied three categories. First one is bacteria, the other one is cyanobacteria, and the third one is mycoplasma. This third category does not have cell wall. Both these categories have cell wall. Now let's talk about the genetic material present inside prokaryotes. They do not have a well-defined nucleus, and in place of nucleus, they have nucleoid. Nucleoid contains the genetic material which is naked. Naked does not means it does not wear clothes. In terms of cellular biology, naked means not covered by a membrane. So the genetic material is naked in prokaryotes, which is not enveloped by a nuclear membrane. Now let's discuss about the plasmid. What do you mean by plasmid? Plasmid is a type of DNA which is present outside the genomic DNA. The characteristic features of plasmid is that it is circular, it is smaller, present in the cytoplasm, extra to the genetic material. So for bacteria, in addition to genomic DNA, the genomic DNA stands for the DNA which contains DNA, circular DNA, or the chromosomes. The single chromosome which a bacteria has is called as genetic DNA, and many bacteria have these plasmids which are small circular DNA which is present outside genomic DNA. So let's see a typical bacteria. So bacterial DNA, which is the genomic DNA which contains all the genetic information of a bacteria, and in addition to this, outside this genomic DNA, there are plasmids. Plasmids are small circular DNA fragments which are present in the cytoplasm outside the genomic DNA. So what are the uses of these plasmids and why they are present in a bacteria? So let's study this. Plasmid DNA confers unique phenotypic characters to the bacteria. So we have two types of characteristics. When we'll be studying about the genetics, we'll see there are two types of characteristics each organism has. One is the genomic characteristics and the other one is phenotypic. Genetic characteristics are like the height, weight of anything, whereas phenotypic characteristic is how the thing looks. What are the looks of that particular organism, and other features which makes it unique in comparison to other organism. For the time being, in bacteria, some bacteria have resistance to the antibiotics. That is why we have a wide variety of antibiotics, because few antibiotics work on few organisms, others will work on other organisms, and similarly each bacteria has inbuilt resistance to a particular type of antibiotic, which is provided to that bacteria by the plasmid DNA. So this plasmid DNA provides unique phenotypic characteristics to bacteria, such as resistance to antibiotics. And this most important line which you need to remember is that plasmid DNA is used to monitor bacterial transformation with foreign DNA. This statement is from biogenetics, that is engineering done on DNA, in which we provide a particular type of DNA to a bacteria which we want it to have it in itself. For example, if we want E. coli to form insulin as per is required for the diabetic patients, so we need insulin to be produced in large amounts. So we will take that gene which is encoding for insulin and we will insert that into a bacteria which has high multiplicative rate, for example E. coli. So we want that gene for insulin to be inserted into the DNA, genomic DNA of E. coli. So we will use a plasmid which will use as a vector, which will use as a transporter of that favorable gene, which is known as target gene. In my example, take the example of insulin. So for the gene of insulin, we have taken a plasmid in which we have inserted the target gene, and we know that plasmid DNA has certain antibiotic resistance gene in itself, which provides a unique phenotypic characteristic to each bacteria. So once we provide this plasmid into the bacteria and heat that bacteria, the tendency is that this bacterial DNA, genomic DNA, acquires this plasmid DNA when heated. And to check whether the plasmid DNA is acquired by the genomic DNA, we put that particular bacteria into the gene, into a medium which has that specific antibiotic whose resistance gene was present in the plasmid. If the bacteria survives, this means plasmid DNA has successfully merged with the genomic DNA, and if the bacteria dies, this means plasmid was unsuccessful in merging with the genomic DNA. So this tells whether the bacteria has transformed with the foreign DNA or not. Understood? So we'll be discussing this in detail in class 12th. So now let's see what are mesosomes. Mesosomes are characteristic features of prokaryotes. And what do we mean by mesosomes? Mesosome is nothing special, just an invagination of plasma membrane of the bacteria. That is, it is a specialized differentiated form of cell wall. Differentiated form of cell wall, which is an infolding of a cell membrane. If we have this as a cell wall and this as a cell membrane of a bacteria, so at some places this would invaginate into the cytoplasm and would be differentiated to form mesosomes. So three important things you need to remember is, what is mesosome? Mesosome is specialized differentiated cell wall which has invagination into the cytoplasm and is a characteristic features of prokaryotes. Now let's see what are ribosomes. Ribosomes, as we know, are the protein factory of the cell which synthesize proteins, and these are non-membrane bound organelles which are present in both prokaryotes and eukaryotes. And one thing special or say unique to prokaryotes is inclusion bodies which are present only in prokaryotes. So inclusion bodies are present only in prokaryotes and are unique to prokaryotes. Now let's discuss in detail the cell envelope of a bacteria. So what is a cell envelope and what is its function? Each bacteria is covered by a layer called as cell envelope, which itself is made up of three different layers, which are glycocalyx, outermost layer, then comes cell wall, and the innermost layer is plasma membrane. So what is the role of this cell envelope? So this envelope protects the bacteria from external damage or external harsh environmental conditions. So for this, most prokaryotes have cell envelope, and particularly bacteria. And this is chemically complex. They have chemically complex cell envelope which is made up of three layers which are tightly bound to each other. The outermost is glycocalyx, which we'll be discussing, which will have two types based on the consistency. Second layer is the cell wall, and the innermost layer is the cell membrane. So let's see the diagram showing this. So first of all, let's make the three layers. The outermost is glycocalyx. Glycocalyx is of two types, slime layer and capsule. So this capsule is telling us that this is a part of glycocalyx. Then comes the second layer, which is cell wall, and the innermost layer is plasma membrane. So we can see this in diagram. Capsule is described as this blue layer. Yellow layer tells us about the cell wall, and the innermost layer, which is green, is a plasma membrane of the cell. These three layers together form the cell envelope, which is present in most of the prokaryotes and especially bacteria. Now let's see the function of individual layer. Each layer performs specific function, and together they form a protective unit for the bacteria. They protect it from chemicals, from external environmental conditions, and also see on the basis of this staining of cell envelope, we have categorized bacteria as gram-positive bacteria and gram-negative bacteria. Those bacteria which take up gram stain are called as gram-positive bacteria. They take gram stain and are purple when seen under the microscope. Some bacteria which do not take gram stain are called as gram-negative and are seen as pink-colored bacteria under the microscope. Now let's discuss in detail the individual layer of cell envelope. There are three layers. First, an outermost layer which is called as glycocalyx. It differs in composition and thickness among different bacteria, and depending upon the composition and thickness, glycocalyx can be of two types. If it is loosely arranged sheath, it is called as slime layer, and if it is thick and tough, it is called as capsule. In the diagram which we have discussed right now, we have a capsule. This means this layer is thick and tough. And if this layer would have been as a loose sheet, this would have been called as slime layer. So these are the three layers, and we are discussing the outermost layer, glycocalyx, which differ in composition and thickness among different bacteria. If it is loose, then it is slime layer. If it is tough and thick, it is the capsule. Now comes the second layer of cell envelope of a bacteria, which is cell wall. The functions of cell wall is it determines the shape of the cell. It provides structural support to the cell, and the final most important thing is it prevents it from bursting. Now let's see what does this mean. If we have a solution which is hypotonic, that means the solutes are less in this solution, and if a bacteria is placed in this solution, what will happen, water from this solution would pass into the bacterial cell, by the process called as osmosis, and this bacterial cell would ultimately swell up and ultimately burst open. But due to the presence of cell wall, what happens is that this bacterial cell resists the increment in its size just because of this cell wall, which provides structural support. Bacteria does not enlarge this much to a level in which this will burst open. So therefore cell wall prevents the bacterial cell from bursting. So three important functions of cell wall, which is the second layer of cell envelope of a bacteria, is that it provides support. Second thing, it provides shape, and third thing, the most important thing, it prevents the bacteria from bursting and collapsing. The innermost layer is the plasma membrane. Plasma membrane is semi-permeable in nature, and similar structure as that is present in eukaryotic cell membrane or plasma membrane. Same plasma membrane is present in prokaryotes. So we have discussed the three layers of cell envelope of a bacteria. Now let's discuss the mesosomes. We have already discussed mesosomes, three important characteristics. First one is, it is a characteristic features of all prokaryotes. Second thing, it is specialized differentiated cell membrane. And third thing is that it invaginates into the cytoplasm. Now let's see the structural details of mesosomes. It is a special membranous structure formed by the extension of plasma membrane into the cell. See this diagram, we can see that we have a green colored cell wall and a yellow colored plasma membrane, and this plasma membrane is invaginating into the cytoplasm to form a specialized structure which is known as mesosome. So these extensions can be of three types. Either they can form spherical structures called vesicles, or elongated tube-like structure called tubules, or plate-like structures called lamellae. So mesosomes can have three structures, which are vesicles if they are spherical, they can be tubular if they have tube shape, and third thing is they can be lamellar if they have plate-like structure. There are 5 important functions of mesosomes. So let's discuss them individually. First thing is cell wall formation. When there is cell wall formation in a prokaryote, these mesosomes helps in the establishment of cell wall around the plasma membrane. Second important thing is they help in DNA replication and distribution of DNA to the daughter cells. The most important function is respiration. They have enzymes which are required for the respiratory processes, and inside prokaryotes most of the respiratory process occurs in mesosomes. Fourth important thing is they help in secretory processes. They can secrete important metabolites into the cell, and also they increase the surface area to perform more enzymatic reactions. So let's recap the 5 important features of mesosomes. The first one is it helps in cell wall formation. Second thing is it helps in DNA replication and distribution of DNA to the daughter cells. Third thing is the most important thing, it helps in respiration. The single most important thing which a mesosome does is help in respiration. Now let's talk about the fourth point, it helps in secretion of metabolites. And fifth one and the last one, it helps to increase the surface area to provide more space for enzymatic reactions. Now let's discuss chromatophores. Chroma means color. Chromatophores are specialized membranous extensions into the cytoplasm. Just like mesosome, they are also extensions into the cytoplasm which are present only in some prokaryotes, whereas mesosomes are present in most of the prokaryotes, especially in bacteria. Chromatophores are characteristic features of cyanobacteria. They contain pigments. Chroma means color, and color is given to the bacteria in the terms of pigments. So 3 important things about chromatophores is they are also specialized invagination into the cytoplasm. They contain pigments, and the third thing is they are present in some prokaryotes like cyanobacteria. Now let's discuss the structural details of flagella. Flagella. Some bacteria move whereas other bacteria are stationary. The bacteria which move due to the presence of flagella. So let's see what does a flagella contain. Bacterial cells may be motile or non-motile, and if they are motile, they have flagella. Flagella is a filamentous extension from the cell wall. It is an extension from the cell wall, not cell membrane. Now let's see that bacteria have a wide variety in the number and arrangement of flagella. For example, let's see this diagram. Some bacteria do not possess flagella. Some have only single flagella. Some have two flagella on both ends. Some have many flagella on both ends. Some have flagella all around their body, and some have flagella only on one end. So there are wide varieties of bacteria on the basis of number and arrangement of flagellas. The flagella of bacteria contains three parts. The longest one is called as filament of flagella, which extends from the cell surface to the outer surrounding. In the diagram we can see this light colored structure, which is filament. The second structure is hook of the flagella, and the third is the basal body of flagella. Flagella arise from the cell wall of the bacteria, which contains three structures. Filament, the longest part, which is extending into the external surrounding. Second part is the hook of flagella, which is shown here, and the third part is basal body of the flagella. So these are the three important parts of flagella. Now with this we have finished the discussion of flagella. Now another outgrowths from cell are pili and fimbriae. What are their functions? Beside flagella, in addition to flagella, some bacteria have pili and fimbriae which do not play any role in motility. Motility means movement. If a bacteria is showing movement, that is solely because of presence of flagella. Pili or fimbriae do not help in movement of a bacteria. So what does pili and fimbriae actually look like and do? Pili are elongated tubular structures which are made up of special proteins. They are tubular structures made up of special proteins, whereas fimbriae are small bristle-like fibers. If you have seen your toothbrush, it contains many bristles over which you apply the toothpaste. So these bristle-like structures, when present on the outside wall of bacteria, are called as fimbriae, and they are sprouting out of the cell. Now what does they do and how do they look like? In some bacteria, they are known to help attach the bacteria to rocks when they are in streams of water, and to the host tissue when the bacteria act as a parasite. So pili and fimbriae have a role of attachment to the bacteria to the rocks when they are present in stream, and to the lining of the organs when they are in host tissue. So let's see the three structures. We need to remember that pili is longer than fimbriae and flagella is the longest one among them. So how to remember that? You can see that pili has one L, that means it is long. Fimbriae does not has any L in its spelling, so this is the smallest one, whereas flagella has double L in its structure, in its spelling, that means it is the longest one. So pili is shown by the brown color in this diagram and fimbriae by small pink colored outgrowths, whereas flagella is the longest pink colored outgrowth from this cell. Now let's discuss about the ribosomes which are present in prokaryotes. Ribosome, being a non-membrane bound organelle, is present in both prokaryotes and eukaryotes. Now it is associated with the plasma membrane of the cell. This is an important point, most important point, that ribosomes in prokaryotes are associated with the plasma membrane of the cell, whereas in eukaryotes it is either present in the cytoplasm or attached with other organelles. For example, it is present inside chloroplast, it is present inside mitochondria, it is present on the surface of rough endoplasmic reticulum in eukaryotic cells. The two subunits of ribosomes present in prokaryotes are 50S and 30S. This S stands for Swedberg coefficient, which defines their sedimentation rate, which is dependent upon the density and size of the ribosome. More the number of S, like 50S means it has larger size and larger density in comparison to the 30S. So when these two come together and form a ribosomal unit of prokaryotes, it forms a 70S prokaryotic ribosome. Do not simply add 50S to 30S and say it would be 80S ribosome. No, this is not a simple mathematical calculation. It is dependent upon the density and the size. When they come together, the overall density reduces and is of 70S. S is the Swedberg unit. So let's compare the prokaryotic ribosome with the eukaryotic ribosome. The two subunits of prokaryotic ribosomes are 50S and 30S, which when comes together form a 70S subunit. Some coaching institutes ask you to remember the subparts of this ribosome, like 5S RNA, 23S RNA. No need to remember this. Just remember the subunits 50S, 30S when combined form a 70S ribosome of prokaryotes. In eukaryotes, the subunits are 60S and 40S, which when comes together forms a 80S ribosome. S is the Swedberg coefficient of sedimentation rate, which defines the density and size of the ribosome. Now let's see ribosomes. We know that these are the protein factory because they synthesize protein. When ribosomes attach to a single RNA, mRNA, they form polyribosome or polysome. We need to know that we have DNA segment. From this DNA segment, via process called as transcription, all organisms produce mRNA. mRNA, with the help of ribosomes, which are the protein factory of the cell, through a process called translation, makes proteins. So on this mRNA strand, when multiple ribosomes are attached, this structure is called as polyribosome or polysome. So this is only told here. When several ribosomes attach to a single mRNA, they form a chain called polyribosome, or we can also call that as polysome. In this diagram we can see this blue colored thing is DNA. From DNA we have formed mRNA. These orange color structures are the ribosomes. When multiple ribosomes are attached to the mRNA, this forms a structure called as polyribosome, or we can also call this as polysome. Ribosomes of a polysome translate mRNA into proteins, which I have described you here. The process is called as translation. Ribosomes make proteins from mRNA. Now let's study about inclusion bodies. Inclusion bodies are characteristics of prokaryotes, and these are like open almirahs without gate. They are present inside the cytoplasm and they store reserved food material, but they do not have a membrane bound structure around them. So let's see, they store reserved material in prokaryotes present inside cytoplasm. They are not bound by any membrane system, lie free in the cytoplasm. So three important inclusion bodies present in prokaryotes are PCG. P stands for phosphate granules, C stands for synophysian granules, and G stands for glycogen granules. So we have three inclusion bodies present in prokaryotes which stores reserve material. First one is starting from P, which means phosphate granules. Another one is from C, that is synophysian granules, and the third one is from G, that is glycogen granules. So just remember PCG for inclusion bodies. Now last topic is the gas vacuoles, which are present in photosynthetic bacteria. Photosynthetic bacteria can be of two types, blue green or we can say purple green. So these two types of photosynthetic bacteria contains gas vacuoles inside them. With this, we have finished the discussion of prokaryotic cells. Hope you like the video. Thank you for watching.